Hello students, let's study physiology of bacteria and culture media.
Hello students, today we will see growth requirement of bacteria. So first is food or nutrients required for the growth of bacteria is proteins, polysaccharides, lipids, nucleic acids, organic, inorganic salts and vitamins. Then moisture is required. Then pH it should around 7.2 to 7.6. According to that acidophilic and basophilic bacteria they are known, classified. Hello students, today we will see bacterial physiology, that is bacterial growth curve. So when bacterium is inoculated into a suitable culture media or liquid media and incubated, its growth follows a definite course and that curve is called as bacterial growth curve. There are four stages in bacterial growth curve. First is lag phase, LAG. Second is LOG, log phase, then stationary or exponential phase, and then log phase or exponential phase, stationary phase and decline phase. In this diagram you can see these four phases. So what is lag phase? So immediately after inoculation on culture medium there is no appreciate increase in number, but there is increase in size of the cells. So this is called lag phase. Log phase, after the adaptive to the new environment, bacteria multiply, and then it is also called as exponential phase. There is increase in bacterial number, bacterial count. Then stationary phase, after log phase cell division stops because of decrease in nutrients and accumulation of toxic products, and then the bacterial count is static. And then after that bacterial count decreases because of the death of the cells, because of the toxic accumulation, and this is called as decline phase. So these are the four phases of the bacterial growth curve. Thank you. Hello students, today we will talk about culture media. So first we will see what is definition of culture media. So culture media is a media required for growth of the microorganisms like bacteria, virus, fungus, parasites, all. So basic, but routinely we use culture media for bacteria and fungi. So now classification of culture media, they are divided into different types. So first is based on consistency. They can be liquid, semi-solid or solid. So in liquid, peptone water, nutrient broth, or in solid blood agar and other all culture media. Based on oxygen requirement, it can be aerobic medium or anaerobic medium. So the bacteria which grow in presence of oxygen, they are called as aerobic medium, and media which do not provide oxygen for the growth of the anaerobic bacteria like clostridium which do not grow in presence of oxygen, they are called as anaerobic media. So first ingredient is nutrient broth, which contains peptone, that is protein part, then meat extract, sodium chloride and water. And if you add agar to this nutrient broth, it will become nutrient agar. So role of agar is to solidify the media. The concentration used is 2%. If you use 6% concentration of agar, then it prevents swarming of the bacteria like Proteus. Enriched medium, here blood, serum or eggs are added to the basic media and it is called as enriched media for the nutritional deficient bacteria which grow in presence of the extra added nutrients. So examples are blood agar, chocolate agar, Loeffler serum slope and egg based media, that is Lowenstein Jensen medium. And then if more than one type of bacteria are present, special substances are added to favor the multiplication of particular species of the bacteria or to inhibit the other bacteria. So they are called as selective media. Examples are selenite F broth, tetrathionate broth, alkaline peptone water, bile broth. Selective media, selective agents are added to solid the media and which act as an enrichment, but they are solid in consistency. So they are bile salt agar, deoxycholate citrate agar and TCBS for the Vibrio. Indicator medium, indicator in the media changes color when particular bacterium grows in them. So example of that is blood potassium tellurite agar, Wilson Blair medium. So blood potassium tellurite agar is used differential media which differentiate the colonies from one to another. Example is McConkey agar. So on Some delicate bacteria, they need transport media, so it is towards medium or Venkatraman Ramakrishna medium for Vibrio, Carrie Blair medium for Salmonella, Shigella, Vibrio, etc. Anaerobic medium, so the bacteria which do not grow in presence of oxygen, so media provides deficiency of oxygen in that medium and they are called as anaerobic medium. So they are Robertson cook meat media and thioglycolate broth media. Some media are there for the growth of the fungal like Candida, Aspergillus, Penicillium, Rhizopus, Cryptococcus. So they are the ideal culture media for fungal isolation is Sabrot's dextrose agar, and dextrose is 4 gram, neopeptone 1 gram, agar is 1.5 gram percent, and distilled water. So here the pH is 5.4, and then because of that other commensals are not grown to this acidic medium. The antibiotic sensitivity testing media is done on Muller-Hinton agar for the antibiotic sensitivity testing, and that is usually done by Kirby-Bayer disk diffusion method. Media. So sticking on the plates is done this way. So it is first you have to take this, you have to heat the wire loop on flame till it becomes red hot, then wait for some time so it will be cool, and then after that you have to take the sample in wire loop and then do primary inoculation. Then from that primary circle you have to draw the lines horizontal, then rotate the plate, then again draw the lines, then rotate the plate, again draw the lines. So this, and then after that tail in the end. So this is primary, secondary, tertiary and quaternary inoculation you have to do, so that you will get isolated colonies to study colony characteristics.
Hope you find this session useful for your understanding and for your exams. Thank you.